Thank you very much, and thanks for the invitation to speak here. We are a shop that does implementation science and have been doing so around genomics since about 2000, when we first gave back single variants, then graduated to larger panels, then to whole genome sequencing in adults. Along the way, we got involved in designing the secondary findings return of results for the American College of Medical Genetics and Genomics in 2013. And our history with newborn sequencing dates back to about 2012, where we had been thinking about this as the logical extension of our screening work with adults for obvious reasons. And at that point, to our delight, the NIH, NHGRI, and NICHD put out an RFA for newborn sequencing proposals. And we were fortunate to win one of those and be the only one of those that offered comprehensive sequencing to ostensibly healthy newborn babies. And that's how BabySeq was born. In fact, it was that Time magazine that you showed, Catherine, at the beginning, by Bonnie Rockman, was actually the first public report of our interest in 2013 in sequencing newborn babies. BabySeq has taught us a lot. Let's see if I can click on. It's many people, many more than these have been part of it. So I'm representing a large group of collaborators. Specifically want to call out MPIs Alan Beggs and Ingrid Holm for BabySeq 1 and 2 respectively, and new young faculty, Nina Gold, Nidhi Shah, and Anna Lewis, are doing great work with us on this project. But what's really exciting that I can present to you today is a little bit of real data. And as we think about the real data, obviously we're all interested in the risks, the benefits, and the costs of this technology. And as has been alluded to, screening is very different than diagnostics. But as you'll see from our data, there is a way in which screening segues back into diagnostics in a really fascinating way. So the way we set up our trial was as a randomized clinical trial. And this is, of course, because the kind of people who volunteer for such information are themselves different from historical controls. And this allows us to really parse out some of the medical, behavioral, and economic outcomes in a very rigorous way. And we were able to recruit about 300 families into BabySeq 1. Our very first issues had to do with which genes to report. And with a talented team from the Mass General Brigham Laboratory for Molecular Medicine, we were able to pare down about 1,000 genes. And these were genes that were leaning toward actionability, but not all conventionally actionable. Were really selected for genes with strong and definitive evidence of disease gene validity, because we figured that scientific truth was the most important value here. We also leaned into genes that had, as best we could tell, higher penetrance and, of course, pediatric or adult or adolescent onset. We did end up including a limited number of adult onset genes for reasons that we've published on and I can talk about later. And that turned out to be really fortuitous, I think, in terms of the interest. Now, importantly, because this is often mentioned, we did not decide to return variants of uncertain significance, but only pathogenic and likely pathogenic variants. And I think this is obviously because you want to enrich for prior probability, and you want to try to get as few so-called false positives as possible. Now, it's very interesting that way back in about 2012, we were asking parents on the newborn ward, do you want this kind of sequencing for your new baby? And the vast majority were interested. But when we got down to the 14-page consent form with all sorts of horrible outcomes, issues about privacy and discrimination, and not to mention the fact that we're going to randomize you and put half of you into an arm where you're not going to get any results whatsoever, the enthusiasm was not quite as high. Not to mention that many people, after they've just had a baby, do not actually want to talk to anybody about research. So this is a little misleading, because only 10% of people really said, yes, come on in the room, I'm interested in research. This has been misinterpreted in the media as suggesting that parents don't want this service. And I think that's incorrect. They very much do, and multiple other polls, including the UK Engagement Project, has suggested that. When we did get into the room and talk to them, turns out that about 2/3 of them actually enrolled. So that was a pretty good output. The real surprise in BabySeq 1 was that of the nearly 160 babies that were fully sequenced, 11% had an unanticipated monogenic disease risk. And what was even more surprising was that a quarter of them, when we circled back and examined the baby in light of the DNA finding, they actually had features of the condition that had not been previously suspected. So for example, that elastin mutation you see on the third line can be associated with supravalvular aortic stenosis. That particular baby was absolutely normal heart sounds, no family history, but an echocardiogram in follow-up to this finding did find a narrowed aorta, abnormally narrowed aorta. Now you could say that's a glass half full, glass half empty. Maybe nothing will ever happen about it. But if that baby were to go out on the playground and have a fainting spell, I think this would be a very valuable piece of information to know about. And that's how I'm thinking about so much of this as risk stratification, not as diagnosis. We followed all these babies into their downstream medical consequences, and I won't belabor this. We have a really interesting paper that's up on medRx, a preprint server, if you're interested. But what is extraordinary is how many family outcomes were changed as a result of the baby's discovery of the mutation. We tried to start creating a taxonomy of this using the ClinGen actionability criteria as a starting point. You can see we're playing around here with shapes that might connote the degree of tolerability, disease severity, disease penetrance, and efficacy of intervention. I don't think any of these graphics are really satisfying yet. They don't really shortcut the kind of dialogue we need to have with parents in order to help them understand some of the pros and cons. But it's fascinating to try to think, how can we take these multiple dimensions and create a meaningful and recognizable taxonomy of what it means to screen for different conditions with different kinds of treatability. Obviously, a major question is the economic outcomes, and we're able to show that in the first 6 months after disclosure, those babies who were sequenced did spend more on their healthcare. Of course, that's not really a surprise. What remains to be seen, and what we're trying to answer with long-term follow-up, is the value gained, the discoveries, the mitigation of illness, the security that parents feel in light of symptomatology that emerges. Is all of that a value that is worth the extra cost that will come from sequencing, interpretation, and medical follow-up? We did publish one paper that models simply the cost effectiveness of screening in the pediatric time period for cancers alone. And as the cost of genomic sequencing and interpretation is coming down, this is approaching that magic $70,000 to $100,000 per life year QALY. So, I think this is truly within reach. And by collecting empirical data of this nature, we can start to justify it and lay the groundwork for these larger scale. And importantly, we and others are pushing back on some of the narratives that have obstructed progress in this and related fields. The narrative of catastrophic psychological distress, for example, simply is not true in many of the adult studies we've done, and does not appear to be true here. We never want to be cavalier about the possibility that we give someone bad news or ambiguous news. But on the other hand, the notion that simply telling someone about a mutation that creates some risk stratification of that baby is going to push them into a depression, anxiety, disruption of parent-child relationship, empirically is not supported by our data. So this is a really exciting moment. This workshop, this gathering of minds and opinions and plans. We ourselves are moving from BabySeq 1 to recent funding by the NCATS Institute at NIH for BabySeq 2, where we're enrolling at least 500 babies from diverse backgrounds, and we have additional volunteer sites in the US that may allow us to move that up above 1,000. But importantly for this community, we're so excited to be talking to the other groups today and to those of you who are listening to this so that we can start to harmonize our interventions, our outcomes, and our resources in order to pool and share the kind of data that's going to truly give us insight into not whether we do this, because we all know it's coming, but how we do this properly, effectively, and humanely for the benefit of all humans. So I'm really delighted to have had the chance to speak to you a little bit about our data. On our website, we have now several score papers, including about 24 papers from BabySeq. We'd love to invite you to take a look at there. And I'd be happy to respond to any emails of people who might wish to train with us or collaborate with us as we go forward in this. Thank you.